Hello and welcome to EJC News Focus, coming this month from the TAP Congress in Paris. Dramatic clinical responses have been seen with targeted agents, but their impact on overall survival has been disappointing. Resistance always develops. But could HSP90 inhibitors, which hit multiple oncogenic targets, overcome and prevent resistance? That was the premise of the Congress keynote lecture given by Paul Workman from the Institute of Cancer Research in London. I met him after his talk and asked him first how knowledge of resistance is feeding into drug development.
I've really seen it as a multi-component approach. I think the first one is understanding the mechanisms of resistance and building drug combinations that will attempt to block, for example, the feedback loops, or attempt to overcome the genetic perturbation, particularly 2-drug or 3-drug combinations that will just reduce the bypass opportunities in biochemical feedback loops or block the potential clonal evolution. So drug combinations and thinking about those more intelligently, I think, is important. That's the first point. And I would say, in addition, adding immunotherapy to the mix. Not just targeted therapy, but immunotherapy as well. So that's the drug combinations. Then there's the actual choice of the drug target. Now, a very sobering observation is if you take the number of genes that are responsible for cancer now, some 500 or more, and you look in all the literature at the number of targets which have actually been successfully drugged with a small drug molecule, it's only about 5% of the genome. A cancer genome. So that means that there's 95% of the genes involved in cancer involved in these networks for which we do not have a drug. Now, if we know that cancer cells are going to reset the networks and they're going to have bypass mechanisms and clonally evolve to have different genetic activating systems, we need more drugs to cover more parts of the network. So that's really just saying we have to complete the journey of finding as many drugs as we can that will exploit all the elements of the cancer genome in order to have a chance of properly building the full drug combinations of the future. Now, the final consideration, I think, for drug treatment is that if we continue to make kinase inhibitors, and we should, because we still have some kinases to develop drugs against, we need to think about how resistance will develop and design strategies to overcome that from the very beginning. But also we need to think about if there are targets in cancer biology that may be more suitable for tackling this clonal evolution and network plasticity. Are there targets, maybe epigenetic targets, maybe, as I'll talk about in a moment, chaperone and HSP90 type targets, where you will actually hit the tumor cell at the heart of the evolutionary mechanism. And if we do that, that would give us a particular focus on a new type of drug that might have a chance intrinsically of reducing the opportunity for tumor cells to develop resistance. And we could then add those in to the combinations and be a lot more successful in the future, I believe.
Why do you believe there's a key role for the HSP90 protein?
We always knew that HSP90 would be a target that, unlike, say, a kinase inhibitor, if you inhibit HSP90, you will affect multiple different targets and multiple signaling pathways in cancer. Not so much as for it to look like a cytotoxic drug with a broad blunderbuss type effect, but rather it would concentrate its activities on many of the cancer pathways. So what HSP90 does functionally is it allows, it stabilizes what would otherwise be unstable proteins, allows them to become active in the cell and stops them from being broken down by the refuse disposal system in the cancer cell. If HSP90 wasn't there, these proteins wouldn't survive or wouldn't be functional. Now, why is that relevant to cancer? Because many of the cancer-causing proteins are mutated or present in excessively high levels, and they need the attentions of HSP90 more than the normal proteins, more than the average protein in the cell. So that was the thinking right from the beginning, that if we had an HSP90 inhibitor, we would get broad effects. But really, the experience with HSP90 inhibitors has gone beyond that, just as the recognition of clonal evolution and resistance has developed in parallel. And the two have come together in such a way where we think that inhibiting HSP90 would actually block the evolution of mutated proteins themselves, and thereby block clonal evolution, or at least minimize the risk of clonal evolution. So the thinking goes like this, that first of all, with biochemical feedback loops, if you, say, give a kinase inhibitor, you'll get a feedback loop that will activate the pathway maybe further down the pathway, or will activate parallel pathways, and HSP90 will hit those parallel pathways, and an inhibitor of HSP90 will hit those pathways or hit further down and will overcome the resistance that's due to biochemical feedbacks. If you take the genetic evolution, if you have, as often happens, another mutation that's selected for by a drug treatment, say a kinase inhibitor, and therefore it becomes resistant to the kinase inhibitor, but it still requires HSP90. It might even require HSP90 more because it's had a second mutation, might be even more vulnerable to the loss of HSP90. So we see now HSP90 as having that dual function.
How much progress has there been to date?
They have been shown to have activity in the clinic. We're seeing activity in patients with breast cancer with ERBB2 overexpression who've become refractory to Herceptin, and then you challenge them with an HSP90 inhibitor, and about 25 to 30% of those patients respond to now treatment with Herceptin and the HSP90. And then secondly, we're seeing activity with an HSP90 inhibitor, including one that we've discovered in our institute and developed with pharma colleagues, where we also see now activity in non-small cell lung cancer with patients with ALK rearrangements, which is the driver for them. They've become resistant to an ALK inhibitor. They're then treated with an HSP90 inhibitor, and again we see 25 to 30% response.
So what's the best approach to using HSP90 inhibitors?
Really, if you want to overcome resistance and evolution, you want to use an HSP90 inhibitor much earlier in treatment. Now, two studies that have just come out that support this notion. One is where a model system with estrogen-dependent breast cancer xenografts were treated with either with an anti-estrogen, tamoxifen, or with tamoxifen plus a low dose of an HSP90 inhibitor, and they show there very clearly a much bigger and longer anti-tumor effect, and the inhibition of resistant variants growing out. And the second example is very similar, where you take a melanoma xenograft with a BRAF mutation, and where you treat, again, either with the individual drugs or the combination, and in the combination you get a longer and more profound anti-tumor effect, and you block the emergence of resistant clones. I think those two studies show in experimental models, supporting my hypothesis that the way to use this exciting new class of HSP90 inhibitor is to use them early in treatment in combination with the targeted therapy.
So we can expect to hear a lot more about this class of drugs over the next few years. And with thanks to Paul Workman, that's it for this month's EJC News Focus.